output traits, which means traits for the processors or consumers. Input traits, obviously yield remains a priority. Diseases resistance remain a priority. And field holding also. I put drought tolerance. I might have added water demand or water quality. Earliness is important. I don't know if it is still important in California, but maybe we will know later on. But when I was working on tomato with Nestle in California, I was always told that if we had an earlier variety with a good bricks that could allow to open the factory 1 week more, and getting back to the discussion we had before, opening a factory 1 more week per year was a huge saving of money. Speaking about output traits, bricks remains a priority, but I prefer to call it bricks yield because increasing bricks at the price of yield is not a good solution. Viscosity is obviously a priority for certain category of product, texture, color, lycopene, and I would add flavor, acidity, which is mainly for fresh tomatoes, but which could probably be transferred to processing tomato. The discovery in this domain could be transferred to processing tomato. Now, just few words about the genetic improvement of tomato varieties in the past. I was able to find a paper published 10 years ago in 1999 where they compare the same variety grown during 20 years in California and 20 years in Israel. And as they are currently doing for other crops, let's say corn or wheat, they tried to analyze the average improvement in the two countries for different characteristics. And you could see that for yield, the average gain was about 1.5% a year when it was only 0.5 in Israel. Bricks, they did not have any improvement at that time in California. They had 0.5 in Israel. Bricks yield, you get the data from the two former figures. And they had also some improvement in color. But the main conclusion of this paper, which seems to me quite interesting, was to say that a large part of the genetic improvement came from the replacement of inbred by hybrids. And what has to be mentioned also, and it has been mentioned before, is using hybrids is certainly more expensive for the growers, but it allows the seed and the breeders, the seed industry and the breeders, to stack more rapidly some resistant trait in a given variety, meaning I have these resistant traits in two different varieties, and by producing hybrids, I can stack them quite rapidly. But the main conclusion was to say we need new sources of diversity. And this is where I will enter a little bit more in science, and I will enter a little bit more in the activities of the company I have co-founded with Professor Tanksley in Cornell University, which is how to use or to better use natural genetic diversity. Just for giving you some information about this company has been created 2.5 years ago. It is giving services to the main seed companies on different crops. We are mainly working on field crops like corn, rice, cotton, sunflower, rapeseed, and so on. And we are giving assistance to the seed company for making a better benefit of the natural genetic diversity. So it is clear for everybody that during domestication and breeding, we have lost the breeders and we have lost quite a lot of diversity. And if you look today to the available diversity, you could have a look for many different crops. But if you look to tomato, you could say that cultivated tomatoes are covering maybe no more than 5% of the genetic diversity available in the wild and exotic related species, I would say crossable species. For the reason I have mentioned before, for domestication, for breeding, if you go to sunflower, it is only 2%. If you go to corn, it's 20%. So what does it mean? It means that you have a big reservoir here in green of untapped genetic diversity you could use. But how to use it? And in the past, the breeders, and very logically, and all the breeders did so, were looking for mainly resistant traits in wild species. And if they could see resistance to certain diseases, they use these wild and they cross it with their elite lines and they develop new varieties. Does not mean these wild varieties do not have genes of interest you could not see in the wild plant. And this is what I will try to explain in a few slides and quite rapidly. This is representing the 12th chromosome of tomato. And 20 years ago already in the lab of Professor Tanksley in Cornell, a student, a PhD student, was looking to evaluate how many different genes could drive the domestication from wild to cultivated for certain traits. And they look for the growth rate, and they used cultivated tomato, this one, and they used the wild tomato, they cross it, and the wild is this one. And then, thanks to molecular biology, they could look to the differences for, let's say, growth rate. And they found 11 QTL, as we call them, but loci, say loci to make things simple, which are involved from the wild, which are involved in the growth of the tomatoes. But surprisingly, 9 of them, which are in red, are, no, sorry, 9 of them which are in red are negative, meaning they are driving a low growth rate. But surprisingly, from the wild, from these tomatoes with a very poor growth rate, they discovered two loci, which are encoding for faster growth of the tomato. That was quite surprising. That could have been an artifact, of a statistical artifact, but then it was possible to recombine and to get some lines having only the two blues. And if you look to these tomatoes, this one is the wild, this one is the cultivated one, and this one is the cultivated one into which we have introduced the two positive genes for growth rate. Meaning in the wild type, with a very poor growth, there were some genes of interest for growth, and that was 20 years ago. So then we started the project. Okay. Then we started the project including with Joachim, and we were looking for different genes like this one in tomatoes. And we looked for different traits, and we could get some positive genes from wild tomatoes for all these characteristics, including flavor, viscosity, firmness, acid, bricks, and so on. And two examples very rapidly. That's what we call hidden gene. Here you have a wild tomato called Pampinellifolium. Here you have the original variety, and we could find some genes which are increasing the fruit size. Here you